So so we can put presentations and perhaps recordings. I've not done that, but we'll try and load everything from the meeting today on the website for folks to be able to look through in case they have questions later on or are unable to attend.
I think our first talk. Gil. And if you want to come up maybe over here, I'll try to move this so it picks up how you go. The mic is right there.
Awesome. All right, so good afternoon, everyone. Thanks for coming out. Figured we would just jump right into my project, Early Life History of American Lobster in Coastal Southern New England Waters. So I just wanted to start off by overviewing the Southern New England lobster stock historical landings. As you can see here, we have a time series starting in 1982 that runs all the way to 2018. And on the y-axis, we have landings in millions of pounds. You can see in the 1990s, the lobster stock in Southern England was particularly robust for the area, hitting its peak landings in 1997. And sort of in the early 2000s up until now, there's been an abrupt reduction in the populations as evident through the landings. And this is due to a combination of a multitude of factors, including fisheries mortality, as well as some environmental changes, and the onset of an epizootic shell disease outbreak that occurred in the late 1990s. Sort of underlying all of this is a general climate change problem where warming oceans are putting stressors on these lobsters, which are already in sort of the southern end of their geographic range. So just to take a look at recruitment year for lobsters in southern New England, this table here shows spawning stock biomass on the x-axis versus recruitment in millions, which has been lagged to match up with a specific year. And this is a time series that starts in 1979 right up here and follows all the way down to 2010 at the bottom. So you can see in 1997 and 1996, the years that we were talking about having its highest landings, the spawning stock biomass was particularly high, coupled with relatively successful recruitment. And then what's So what's concerning about this is in the early 2000s, when we see that decline in population, we see that there's sort of a decoupling between recruitment and spawning stock biomass, where recruitment is failing, biomass is sort of staying the same and maybe decreasing a little bit as we travel through the 2010s. So this sort of tells us that the lobster stock is experiencing a recruitment failure, and that there's a population bottleneck occurring somewhere between the egg stage and when these lobsters are recruited into the spawning population, whether that be in the larval, post-larval, or juvenile stage. So that sort of leads us into the project objectives, which are to understand and quantify that recruitment bottleneck, sort of figure out where in the lobster life history stage that this is occurring. And to do that, we'll be assessing the abundance and spatial distribution of larval lobsters and post-settlement lobsters in southern New England. And some of the sites we've chosen to do this align with historical sites, so we can test the abundance and spatial distributions we find with historical data to sort of observe how these things are changing through time. So just a quick overview of the life history stages that we'll be focusing on. So the first one here is the larval stage. So after lobsters hatch, they spend about 3 to 12 weeks in the larval stage, and within that, there's 4 separate stages, 1 through 3, and then the post-larva, or stage 4. And once they usually do this in the summer. This is summer is when the stage takes place. And once they're ready as post-larva, they'll actually settle down to the benthos. And this is the post-larval settlement stage. And then they'll spend the rest of their life down on the benthos, where they'll grow up in about 6 to 8 years and join the fishing stock size. So those are the 2 sort of life stages that we'll be focused on in the study. And to quantify the abundance and distributions of them, we'll be doing 2 different sampling techniques. For the larval sampling, we'll be using Newston net tows, towing in the top of the water column, looking for larval lobsters. And that can be seen on the left there. And then for looking for young of year and juvenile lobsters, we'll be using post-larval benthic collectors that are passively resting at the ocean floor throughout the summer. So I'm just going to go over some of the field work that we did this summer to start with the Newston tows. So we had 6 sites in Rhode Island Sound and Narragansett Bay. It's indicated with the green points on the map there. And we were sampling weekly from June 1st to August 1st. There was a slight technical difficulty with the engine motor, so we missed a few days in mid-June, but we ended up getting 6 sample dates. And each time we went out at each site, there were 2 15-minute net tows that were conducted traveling about 3 knots. And we were using a Newston net with about a 1,000 micron meter mesh. And then attached to the net was a flow meter, and this flow meter allowed us to calculate the volume of water that was sampled, so we can get an abundance per volume of larval lobsters. And so after we collected the samples on the boat, we put them in jars and preserved them with 4% formaldehyde, and they were returned to the lab for further analysis. And at each of these sites, we also collected environmental data using a CTD, so we can sort of get a picture of how the temperatures are changing at each site throughout the sample. So while that was all happening during the summer, our benthic collectors were already down there. We deployed them in May. And so we had 10 sites, and these sort of overlapped with the larval sites. So we could compare both the larval abundance and the post-larval settlement indices, as well as some of the sites overlapped with historical locations and some DEM suction sampling sites. And so at each site, there were 10 collectors in a trawl. And the collectors were filled with cobble to simulate a suitable larval lobster habitat post-settlement. So in the bottom right there, you can see a schematic of very simple schematic of what the benthic collectors look like. And there's 2 different types. There's the traditional schematic, and then there's the new version. So you can see half the sites we used this traditional collector, which had larger cobble in it, which provided ample habitat for lobster settlement. The second 5 sites, we used a mix of traditional and new collectors. And the new collectors are a mix of the cobble and smaller gravel. And this was a recommendation by lobstermen who seemed to suggested that maybe smaller young of year lobsters that have just settled seek refuge in the smaller hiding places provided by the gravel. And at the bottom of underneath the rocks, there's 2 millimeter mesh, and that will, the 2 millimeter mesh allows for when we retrieve the collector, all the organisms will stay put. So as we're lifting it up, we can collect all of the organisms in the benthic collector. And so the larger organisms were measured and released, and everything else was bagged and frozen and brought back to the lab. And we'll be doing some further analysis on that. So for both the Newston and benthic collector samples, the lab analyses are sort of ongoing, and we're still working through some of those, but I figured I'd just update you on what's been going on so far. So for the Newston samples, we've been taking them back to the lab, and we're sorting through all of the debris and seaweed and things. And we're sorting the crustaceans, fish, and other zooplankton out, and we're preserving them in 70% ethanol. And the crustaceans and fish will be identified to species, and they'll be counted. So we'll get count data for those. And then the lobster larvae will also be counted, but they'll be identified specifically to their stage. So stages 1 through 3 and post-larva. And I've included a couple pictures here of what we found so far in the preliminary sorting. So on the right, you can see these are some larval lobsters we've collected. The post-larva on the top of stage 3, below that. And then there's some crab larvae, crab megalope, and this is actually a conger eel, leptocephali, that we collected. So while that's going on, we're also looking through the benthic collector frozen samples that we brought back. And some of the collection data was done actually on the boat while we were sampling. And so in the top middle, that's a settler lobster that we caught in the collector this year. And so we're going through these bags and sorting out the crustaceans and fish and measuring them as well. And some of the species that we've been running into is a little more diverse, but we've got black sea bass, cunner, a lot of Asian shore crabs, cancer crabs, and we even pulled up an octopus, as well as a short big eye, which is a tropical fish that's a little far from home. So currently, we don't have any of the abundance data for the larval lobsters or for the benthic collector settlement indices because we're still sorting through all of those samples. What I can present to you data-wise is our flow meter calculations, just the volume of water that was sampled during our Newston net tows. And so in the top right, you can see these are the equations we used to calculate how much volume was sampled. And this is assuming that, as you can see in the picture, the net actually rides about 50% of it's out of the water. So the net dimensions, we just cut that in half. So you can see that we've gotten pretty consistent volumes for each of our sample dates, which is a good sign. And we'll be using these volumes to sort of quantify the abundances of larval lobsters per 1,000 meters cubed. And it's good for consistency's sake that we sort of stayed in the general same range for the volumes. And just wanted to go through kind of a summary and some of the future steps that we'll be taking. So for the benthic collectors, we ended up finding 13 juvenile lobsters in the collectors. One of them was classified as a young of year, which was less than 12 millimeters carapace length per Wally et al. In 2013. And another one was very close. I think it was like 13 and a half or something. So we did find some juvenile lobsters in the collectors, and we have identified some stage 1, 3, and 4 lobsters in our Newston tows. And so as we continue to sort through, we'll be able to identify, measure, and count more of the species. And the goal is to sort of compile that count data to determine the larval abundances at each site, and then as well as the settlement index for each specific site as well. And so that was the sampling for 2021. We do have another sampling year planned for upcoming summer in 2022. And so we'll be planning on Newston sampling again weekly from June to August, and deploying our benthic collectors, restoring them first and deploying them again in May through September. And some things we're thinking about is, maybe potentially targeting deeper sites with the collectors, in addition to see if these lobsters are seeking thermal refuge at deeper sites, and also potentially investigating some offshore habitats. And then those are my references that got me messed up. I'd like to thank Jeremy and Connor, as well as Al and Corinne and everyone for helping out, and I'd be happy to answer any questions.
One question, and then a comment. Why was the 15-minute tow selected? What's the time?
Yeah, so 15 minutes, I think, was determined. I forget where the source is specifically. It was determined that it was enough time to sample a significant amount of water to find larval lobsters in abundances that could be used for sampling, and we coupled them together with 2 15-minute tows just to sort of be able to test replicates to see, if we did maybe 1 30-minute tow, if that was an aberration, the two will allow us to sort of test the results against each other to see if there's randomness involved.
Any idea how long a distance that tow would operate? If you towed it for 15 minutes.
Like just like if you're straight, how far you go?
Yeah. Exactly.
So I don't have. Let me go back to the calculations. So part of those equations allow you to calculate the distance. Yeah, at the top, the distance in meters. So I don't have those specific distances offhand, but basically what we're doing is we're calculating the distance and the speed and using that to figure out how much water we actually pull through the net.
I guess I just add to Declan's comment. that it's a balance between how long you tow, make sure you capture the species, particularly in low abundance like maybe lobster larvae, and then avoiding clogging of the net. You tow for too long, especially with the new stock and seaweed at the surface, they're pretty susceptible to clogging, and then you're not necessarily filtering water.
Okay, one comment I'd like to make. You selected a lot of the sites for roots until parallel where we set the collectors. Would it be viable to make some tolls in other areas so you can have a comparison between the two areas?
Yeah, definitely. I think that especially where we have the collectors down, because I know we have collectors at more sites than we are doing the Newston sampling, it would be beneficial to sort of have both the larval Newston data and then have the collectors right beneath it to see if we're finding, larval lobsters at the top and at the bottom. Maybe there's no problem between the settlers, finding suitable habitat.
In addition to that, selected further sites away from where the collectors were. It could give you some kind of a relative comparison to make.
Oh, yeah, I
The
see what you're saying.
abundance, the larvae.
Yeah, definitely.
I mean, that would just be a suggestion on my part. I was gonna do it, I'd like to set it up like that,
Yeah.
Just a thought,
No, yeah, that's a good idea. Thank you.
future reference. Okay, thanks.
Do you know why you didn't see any ideas why you didn't see any stage 2 larvae? Is that a short stage or are they really deeper? Or?
Yeah, I'm not entirely sure. I think I was expecting to find more stage 3 and 4 lobsters. And at the beginning of the first tow that happened on June 1st, there were more stage 1s, and almost there was no post larvas at all through that entire sample day. So I'm not entirely sure why there were no stage 2s, but it was just, we've done a preliminary identification. So once we sort through all the samples, I'm going to go back and double check that I've identified each stage correctly. So, go through and check again to make sure we didn't miss any stage 2s in there.
All right, I see we have Rich Bell has his hand up. I'm going to ask you to unmute and you can answer or ask your question.
Excellent. Thank you. Interesting work. And I have a question. I'm not positive you can answer it, but I was curious if you had any sense of whether 13 lobsters from your benthic collectors was a high or low number, or if you had any comparison between work done previously, either in Southern New England or Gulf of Maine. And then I was also curious if the benthic collectors, if you had any plans or if it was possible to compare that to some of the recruitment indices that I think with the vacuum suction samplers they are doing, like Connor's out there scuba diving doing. I'm just curious if those can be compared at all, if there was any plans that once you have some more data to be able to compare them or not. Thanks.
Yeah, thank
If
you.
you wouldn't mind, could you just reiterate the question? I'm not sure the speaker's coming through very well for everyone online.
Yeah, definitely. Yeah, so the question was, with the 13 lobsters we found in the benthic collectors, if there's something to compare that to with either research done in the Gulf of Maine or historical studies to see if that was a significant value or if it differs a lot from the other studies. And then the second part of the question was, well, I guess I'll address the first part. So it's just from my understanding, it's not a very high count for what they especially are finding in the Gulf of Maine, but even what has previously been found in Rhode Island Sound. I'm not sure specifically the numbers, but Kristen Clement, who helped do the benthic collectors with me, she works up in the Gulf of Maine. I think she had mentioned that they average about 1 lobster per collector. We were averaging about 13 per 100 collectors. So there's definitely a discrepancy between what we're finding here and what they find up in the Gulf of Maine. The second part of the question is whether or not the benthic collectors that we use can be compared with suction sampling surveys that the DEM has been using. And Connor, I think you know more about this than I do, but I would say yes, they can be used to compare to the suction sampling. It's based on how many lobsters you catch per meter squared. And so I believe that they're just 2 different techniques and there's pros and cons to both, but they can be compared to each other.
Yeah, thanks, Rich. So yes, we'll definitely compare them. I would also say that all the larval lobster stations are where we do the suction sampling for DEM. And then I believe 2 or 3 of the 10 collector stations were picked at concurrent settlement sites for suctioning for that reason. Like over the 30 years that we've been doing it as part of the American Lobster Settlement Index program, we've been trying to collect data on the side-by-side methods comparison. As Declan mentioned, there's pros and cons to both, and there's theories as to which ones may select younger year or juvenile lobsters better than the other. So this data will add to that growing data set of the comparison between the two methods.
Yeah, I just want to point out one thing too, Quan, is you do the sample selection, that's basically in very slow water, right? So our collectors will set in deeper water. Sure. So it's, I would say it's going to be tough to make a comparison except for a few sites with the depth is similar.
Yeah, so that's a good point, Al. So yeah, so some of the sites we picked for that methods collaboration, and the other ones we picked because we have no data from deep sites, so we have no idea whether we're missing parts of the settlements component of Rhode Island by just looking at shallow stations. So each of the sites were picked to help crosswalk us through various aspects of early life history sampling for Rhode Island. So yeah, good point.
Not seeing any other hands up. Anyone else here?
Well, now further to your question about the Houston sampling, we were towing at about 2.68 knots.
Yeah, 2.5 to 8. Yeah.
But for 5 minutes, we're going half an hour per mile. We don't go in a straight line. Don't.
You don't go in a straight line?
No, don't.
Oh, okay.
Kind of do donuts around the.
I see. Okay.
And I wanted to ask you whether you had specific ideas of where to sample for the Houston, or whether you were just in general saying try some other areas.
Yeah, I would like to see some other areas, try to be honest with you. I mean, I could sit down each day, discuss it with you. But
No, we could do that as we plan next year. The thing I point out is there's a logistical thing about we're trying to go out once a week. And so we have a sort of comfortable amount of stations we can do. You can see we're spread out from our refuge over to Fabulous.
Yeah, right.
I think that
Yeah, it's good.
we can do that on a good day, but I'm not sure.
Could you incorporate more days in the research? Do some polls?
Theoretically,
I'd just like to see a little bit broader area sample, see if there's any differences.
I appreciate that.
I'll say too that the 6 sites we did again with the settlement because Rick Wally and Stan Cobb did those sites for 8 years in the 80s and 90s. So it allows for site-to-site comparison across multiple decades to see what they saw at the Harbor of Refuge was for lobster larvae with different than what we're seeing now. So that was part of the idea of going back to the suction sample site as well. Notwithstanding the need to sample more broadly, but that was the original basis.
You're talking about the nuisance codes?
Yeah.
And in Declan's defense, I'll say we have a lab full of seaweed.
Also with the collectors.
Yeah, right, yeah.
Thank you.
No hands up online.
One more quick question. I'm sure you're going to probably discuss this in the future, but is there any thought given to increasing the amount of collectors we have, different size box in them? Because this year here, we only did, what, 25 out of 100? See, maybe 50 out of 100 myself personally. Because I think, if I remember correctly, most of the losses were caught in mixed box. So it just makes sense to me that a few more of those, maybe up to 50% instead of 25. It was a pilot program, I realized that. After seeing what the results were, I would suggest that we increase, like say 25, 50 collectors, mixed lots. It would just be my thought.
Have you done that analysis, Declan, about the odds of capturing the traditional versus the mixed?
No, I haven't done like the full analysis on it, but I will say to your point that the two potential younger beer lobsters, the one for sure that was under the one that was like 13.5, they were both found in the gravel combination. I think of 13, I don't know the number off my head. I'd say the smaller ones were definitely found in the gravel. 6 to 7 were also found in the normal ones too. So, once we sort of pick through the rest of those samples and get it solidified with where we found the lobsters, I think it's a good suggestion to look back at maybe changing which of the traditional versus new collectors.
I think that small one was 8.5, if I remember correctly.
Yeah. 8.5.
It's the smallest one I've ever seen.
Yeah. Fortunately, I don't have a picture. That was cool. Very small.
There's something to think about, going forward
Yeah.
next year, if you guys want to think it over. Decisions you have, it's just a recommendation.
No, we're definitely going to convene a team meeting where we start next spring to get your.
Stefan, I guess my last question is, I know you haven't done the estimates yet, but do you feel that starting in June captured lobster larvae adequately? Did we come in at high abundance, or should we start earlier because of like when the eggs are being hatched? I know that's, or maybe it's just a question to think about when you look at the data seasonally over the 6 days. If it's constant at some abundance, maybe we want to start earlier to catch the ramp, or it seems like, if I remember correctly, by August we saw none. So that kind of instigated the cease of the program then.
Yeah, so that's a great point. And there's a little hiccup in that because the first week, actually Corinne helped sample the first week in June, and then the next 2 weeks we missed because of the boat incident with the motor problems. So it was kind of hard to gauge because we had one day in June and then one day at the end of June. So it was a little difficult to gauge if it was either ramping up or we just jumped right into the sampling. But I would say, based on what I've seen in the literature for the length, how long they see the larval lobsters, I think that June 1st, we didn't see any post larvae, and sort of from early June to the beginning of August matched up with how long estimates were or what they would be. Definitely something to consider, but I think June 1st is probably a pretty good day to start.
I have a question in the chat from Greg. Are there any signs of predation events taking place within the benthic cages, i.e. small claws, etc., being found?
It was a little hectic when the benthic collectors came up because there was a lot of seaweed and settlement and a ton of Asian shore crabs and things like that. But we did find a decent amount of small black sea bass and cunner, which are known to predate on small lobsters. So there were definitely predators of lobsters found in congruency with them in the collectors. I don't think we had ever found evidence on a specific lobster that it had been that there was attempted predation, but definitely evidence of predators.
All right, nothing else online.
Thanks so much, guys. Appreciate it. The only problem with my presentation is it sort of flows with my.
I saw. I'll try to move
Okay.
forward as you're talking.
Yeah. Alright. Is there any way that I can put through it?
I'm trying to monitor both this and this.
Okay,
I have to
yeah,
jump
sure.
back. Otherwise, I would have you just do it.
Okay, so thank you everyone for being here. My talk is on the response of benthic fauna to changing environment in area today, which I have subtitled, which is a question I get very often on my research is who really cares about worms? Next. So I think the best way to start the talk is to take apart the title. So again, the title is Responsive Benthic Fauna to a Changing Environment in Narragansett Bay. So my first slide will be on what is benthic fauna. How is the environment in Narragansett Bay changing? What is the response of these critters to the changing environment, and why do we care? Again, who really cares about worms in a fisheries conference? So what is benthic fauna? When I refer to the benthos, it is the bottom of the water column, so anything that lives on or below the sediment. And this is actually a very biodiverse group. You will find several phyla that are represented down here, including worms, arthropods, mollusks, sponges, cnidarians, echinoderms, and demersal fish. There is no sunlight that reaches the bottom, although there is very strong benthopelagic coupling, especially in the Narragansett Bay estuary. There is no photosynthesis at this level. So everything that is down here is respirating, but there's no oxygen that's being balancing it from photosynthesis. So everything is fueled by either predation or marine snow. And that's anything that's coming from the water column, so carcasses and feces. And so just as a terminology review, epifauna is everything that lives on the sediment, and infauna is everything that lives in the sediment. And macrofaunal infauna is everything that's above 500 micrometers. So how is the environment in Narragansett Bay changing? So the short answer actually is hypoxia, but it's a synergistic effect between both stratification and eutrophication. And so what we see is because of climate change, especially in the summer, there's increased sea surface temperature and increased freshwater input, especially in the upper bay. And so when you have this huge density difference between the top layer and the bottom layer, there isn't enough turbulence to overcome that density difference. And oxygen is not mixing like it normally would from the air-sea gas exchange. On top of that, you have eutrophication. So this is an increase in the rate of nutrient loading into the bay. And again, this is especially happening in the north of the bay because you have a lot more human development in the north of the bay. And so what this does is it overloads primary production, which also fuels secondary production, but it is unbalanced. So a lot more respiration is occurring than can be balanced by photosynthesis or air-sea gas exchange oxygenation. So these two combined effects, and as I mentioned, the north to south gradient of human population and impact. So what you have is much more in the north, you have the nutrient loading, and you also have much more freshwater input. And you see a lot more impermeable surfaces, so there's a lot more runoff. Whereas in the south, there's a lot less human development, there's a lot less freshwater input, and there's a lot less nutrient loading. Now, in Greenwich Bay in 2003, there was a massive fish die-off, and this was caused by an anoxic event from several rainwater events that happened. And so the combined effect of stratification and eutrophication was suspected to cause this anoxic event that killed millions of methane. And so between 2005 and 2013, the EPA required wastewater treatment facilities to decrease nitrogen input into the bay by 50%. Next. So while this was going on, this mitigation of the eutrophication that was going on in the bay, there was also continued increase in the development of the entire Narragansett Bay estuary, and there's continued warming and freshwater input from climate change. So given all of those environmental changes, what is the benthic response? Well, if you think about hypoxia, it actually begins in the sediment. Now, of course, eutrophication and stratification are occurring in the surface, but the effect of that is felt first at the bottom, because sediment is the furthest away from the oxygen source, which is air-sea gas exchange and photosynthesis. And again, because eutrophication is occurring and stratification is occurring, these two combined events prevent mixing of oxygen while there is a ton of respiration still occurring despite oxygen depletion. So the other thing I want you to think about is how much heavier water is compared to air when you think of soil versus sediment. And you can actually see on this guy, this is a nectus incisor. He has what are called parapodia and setae, and those actually move through the sediment for him. And if you think of earthworms, they don't have those. So what they need is legs to actually move through the sediment, and these create tubes that allows oxygen to penetrate deeper into the sediment, and that's called bioturbation. And the problem is when there's no oxygen getting to the bottom, there aren't organisms that are mixing the sediment because they die in the water and the sediment. So the sediment can't be oxygenated. And so the sediment that is oxygenated gets thinner and thinner and thinner until there is no oxygen in the sediment. And this anoxia begins to spread from epifauna throughout the water column. So who really cares about worms? Well, you should. Monitoring benthic organisms is actually a really good gauge for the overall health of the bay, because like I said, hypoxia actually begins at the bottom. So if you want to see how the rest of the bay is going to be doing soon, you want to see what's going on at the bottom. Benthic fauna also have several ecosystem services, including, like I said, sediment oxygenation, tons of bivalves water filtrate. There's nutrient cycling going on because of the sediment mixing, shoreline buffering, and they channel a ton of energy into secondary production into the rest of the food web. So, given all of that background information, what is the objective of the current project? We want to look at long-term benthic responses to the explanatory variables that I have laid out. We want to compare current data to historical data. So the data that we are collecting now compared to data before the 50% decrease in wastewater treatment facility output. We want to cover a wide array of sampling techniques in fauna because, believe it or not, benthic sampling is a little inconsistent. There's a lot of sampling methods that are used. So, and there are also epifauna and infauna. So to cover the wide array, we want to do different sampling techniques. And we also want to incorporate benthic production into food web models. So the field methods that we have laid out were done at the end of June of 2021, which represents peak abundance of benthic organisms in the bay. It is also planned for 2022 in June. And this was done from the Captain Burt. So we collected diver hand-collected cores, and we conducted 2 replicate beam trawls. And you can see from this map where the transects were done on the beam trawls, and you can see where the diver cores were collected. And we had 3 sampling stations, and this actually represents the north-south gradient of the bay. The first one is the Providence River Upper Bay Station, and this represents a lot more of a nutrient-loaded ecosystem. It's historically a lower successional stage because there's so much nutrient loading. The North Jamestown Mid-Bay Station is historically actually a fairly high successional stage, stable benthic environment, but it also has had an effect from the decrease in wastewater treatment facility output. And the last one is the Rhode Island Sound Station. And so this represents a much more healthy, high successional stage benthic environment, although there may be an effect from climate change at this station as well. So for field methods for each station, we did 15 replicate cores. So the divers went down to the bottom, they shoved tubes into the sediment, they put stoppers onto the tubes, and they brought 15 replicates up for us from the bottom. And using this plunger, well, actually first, we took the overlying water. And it was very, very important to preserve this water sediment interface because there is actually a lot of activity that's going on right there. And these, although they're hard to see in the picture, these are Amphiliska amphipod tubes. And so the overlying water and the top 2 centimeters were preserved separately from the bottom 8 centimeters. And we used the core plunger to plunge out sediment with the measurement of the 2 and 8 centimeter tube. And then also 2 replicate beam trawl tows were done, as you saw on those transects on the map. And so that is the beam trawl net. That's just 2 skis in a bar with the net dragging behind it. And those are all of the organisms that we collected from both the beam trawls. And then all of the samples were stored in both 4% pH buffered formaldehyde and dyed with rose bengal, which turns everything pink. So you're going to see in the pictures that everything is pink. And it's not because everything at the bottom is pink, it's because it was dyed to make identification and counting easier. We also collected YSI data for physical data. So for laboratory methods, everything was taken back to the lab. Samples were sieved through a 500 micrometer mesh, which again is the smallest size for macrofaunal in fauna. And then the organisms were roughly sorted into worms, arthropods, mollusks, and miscellaneous. And you can see from the pictures that it is really easy to see the organisms when they're diving. Now, currently what I'm doing is identifying every organism down to the lowest possible taxonomic group and counting them. So for planned laboratory analysis, once this is all done, we want to take a subset of cores and weigh them and average that for macrofaunal biomass of all of the cores and historical core data. We also want to do pooled weight for epifaunal biomass that was weighed on the boat and in the lab. And from that, we are going to make production estimates. We are also taking 2 replicate 10 milliliter subsets of the course for each station that will be analyzed in Boston University for carbon and nitrogen content. So planned statistical analysis, we have a ton of biodiversity, biomass, and abundance data. So the best way to tackle what we're going to put in for statistical analysis is to revisit the objectives. The first objective was to look at long-term benthic responses to explanatory variables. So if you remember, hypoxia is going on at the bottom, and there are 2 synergistic effects that are causing this hypoxia. And to tease these apart, we can actually use different variables to represent these processes. So for eutrophication and the mitigation, the further mitigation of this eutrophication, we can represent that by chlorophyll data and carbon and nitrogen in the sediment. For stratification, we can represent that by temperature and salinity data and compare those to our abundance, biomass, and biodiversity data. We want to compare this current data to this historical data, especially between the 50% decrease for wastewater treatment facilities. We also would like to find any sort of decadal trends or longer term trends using all of the historical data that we have piled to compare to long-term climate and oceanographic indices. We want to cover a wide array of sampling techniques in fauna because, like I said, a lot of the historical data uses several different sampling techniques. There's also that north to south gradient that I mentioned that we want to cover that array. And there are several different bottom types that are represented in Narragansett Bay. And we're also going to supplement this with analysis of trawl and dredge surveys that have been done by GSO and the Rhode Island DEM. So the last one is, like I said, we're going to incorporate benthic production into food web models. And as I already mentioned, biomass is going to be converted into production for both macrofauna and epifauna. And so I want to thank everyone that has been involved in this project because you cannot accomplish science alone. And I'll take your questions.
Prior to the reduction, the 50% reduction in nitrogen, you said there was a bunch of sampling taken with different methods. How much data do you have gathered before reduction? And were any of those samplings done the same way that you would be doing it now with the tubes, immersion, and sediment?
That is a great question. Actually, yes. So the study that was done right before me by Christopher Calabretta, he did almost the exact same study, and I am replicating mine based off of his. And his was done right before and during the wastewater treatment facility decrease. And there have been several core analyses done in the 60s, 70s, and 80s.
One of the
Please.
What mechanism did the wastewater treatment facilities use to reduce the nitrogen?
What I know is that they weren't, they did not have tertiary nitrate, so they weren't stopping tertiary nitrate from entering the bay. So it's whatever process stops tertiary nitrate from getting into the bay.
You know what that is, by any chance?
I will.
I think it's just microbial denitrification. The microbes that convert the nitrate into nitrogen gas.
But they're using a chemical.
No, it's bacteria do that. Part of their metabolism. Certain bacteria do that as part of their metabolism.
I thought I read one place, 2014, they introduced sodium hydroxide to reduce the nitrogen from the wastewater treatment plants. That's interesting.
Thanks for your talk. I was curious from some of the sorting you've done to date. Can you talk about the species composition that you're seeing in the beam trawl and how that differs from some of the other techniques we're using to monitor the bay? Like
So the beam trawl, what it does is it targets epifauna. So it's everything that lives on top of the sediment. So we're seeing a lot of larger organisms within the beam trawl. So this is sort of like, what it does is it replicates a lot of the dredges and trawls that the DEM does. So it's not the tiny organisms that live in the sediment, it's everything that lives on top. So you see a lot of spider crabs, we saw a lot of whelks, sponges, summer flounder, I believe. So it's a lot of large organisms.
And do you have a sense of how that composition compares to like the GSO trawl or the DM trawl or the?
I have not done that comparison yet.
Following up on that, do you have any feeling for what you think you might be missing?
So that actually, yes. The two things that I've noticed so far, the big one is in Rhode Island Sound. It was cited that a large part of the biomass was Syrian Theopsis, like dwelling anemones. I am finding none of those. So I think that a lot of that biomass was actually replaced by sea cucumbers, which were not historically found, but are now. I am also finding that a lot of the cited sizes, which are the dichotomous keys I'm using are a lot from the 1970s. So what I'm finding is that the size of organisms now is a lot smaller than it was in the 1970s.
For a given species?
Yes.
I see.
We have a question from Marta in the chat. She's asking if you have any data on sea star abundance to this research.
So sea stars were found a lot in the GSO fish trawl up until I believe the 90s, and then there was a large decrease drop off, and now sea stars are no longer found. Although I did have a picture, it's a different species of a sea star than what was normally found, so they are returning. So I did find one.
Not seeing anything else online.
Really good talking. I'm just curious, do you know the species of sea cucumber that you're seeing, or are you seeing more?
Blackea spicrata.
Okay, I'll have to remember that. Just curious, I've been seeing a bunch in Narrow River the past summer. I just wonder if it's a similar species.
Do they have long tails?
No, it doesn't. They're small, gray, and they're kind of round.
Do they have a lot of tubercles?
Yeah.
Yeah, it's probably the same.
It's the same one? Okay, interesting.
We actually have Joe from Rhode Island DEM from the Office of Water Resources answering the question about the wastewater treatment facilities to reduce nitrogen. Each plant uses a different specific process, but they're all using biological nitrogen removal. These processes use a variety of aerobic and anoxic zones in their treatment process to remove nitrogen. So exactly what he had mentioned.
Yeah, that's contrary to the research I've done on it.
Maybe that the paper you might have read may have been a further measure that they had to take. So like the biological process that wasn't working for them, so they just added sodium hydroxide.
You talk about sea stars, just to note, 2014, prior to that, we had millions of sea stars in the bay.
Oh, that's the year?
2014, it dissipated completely.
Yeah, we still don't really know what happened. So it's an interesting point, but I did find one. So I'm not sure if it's the same species. I'm pretty sure.
Yeah, we see one every now and then. It's not every day when we're hauling out here either.
Right.
And basically, it's outside the mouth of Narragansett Bay.
Mhm.
In the bay proper, we see zero. There used to be millions of them around.
Right, and the one I found was in the Rhode Island Sound Station, so it's at the mouth.
And Mark has just commented it's likely sea star wasting disease.
Oh, yes, that's what's been cited. Daniel?
Is any station being fished by commercial boats? So we don't intend.
Question for fisheries expert.
Yeah, I mean, there are certainly people who operate, not in the river, with that river set. I would say most of the trawling is from effectively just south of Quonset in the West Passage, that is, for commercial draggers. But there's other fisheries operating, pond fisheries, cohog, yeah, mantis shrimp is another one. Yep. So there's certainly active commercial fisheries in the West Passage.
Oh, yeah, that's the point.
You talked quite a bit about each petition. Did you look at the profiles that we collected, for example, YSA profiles?
I have not looked at them yet.
Not seeing any hands raised online.
Thank you.
think our next talk is Rich Bell, so you might like for me so it's in front of that because he's going to be speaking through there.
All through the speaker.
Do you think this is going to work?
Fingers crossed. We can hear you. I don't know if online can hear you very well though. Someone online
Okay.
comments for me to let me know if you can hear Rich or not.
So,
I can hear him
yes,
okay.
online will be fine. Yeah. If it's too bad, then Connor could just take over. He's given this presentation a few times as well. So.
All right, I think we're set. So just tell me when to advance slides for you and hopefully this works.
All right, thank you. Well, thank you for being here. And I'd like to say this entire project is actually came out of a RIMFI meeting, originated from a fisherman-derived question, and then really got its genesis at one of the RIMFI meetings, and we decided to make a go of it. So you go to and it grew as the number of co-authors here expanded. So I just want to make a quick note that it started with just DEM, and then TNC, and then brought in Garfo, and then VIMS, and all kinds of people. So we had a lot of people working on this, each doing a small part to make a nice little project. So thanks, RIMFI, for making this all possible. So fisheries dependent data is an essential part of any management system, and it covers, it's necessary for a number of things, from quota management to stock assessments, and even to answer basic research questions. And there are a number of ways to collect fisheries dependent data, from logbooks, VTR, portside monitoring, and a variety of different ways. You go to the next slide. And then in the last decade, a new method has come along called electronic monitoring. Some of you will be very familiar with this. For others, electronic monitoring is the use of a GPS system and cameras on fishing boats to look at what's happening on the fishing boat, usually to count fish. The majority of the time it's used for discards, and it's often used as a complement or potentially even to replace onboard human observers. And electronic monitoring can be very effective depending on the situation. Trying to count every single fish that gets dumped out of a net onto a checker by a video camera, that's kind of hard. But trying to identify a marine mammal that comes up on deck, or trying to measure a slippage event in the herring fishery or something like that, those are perfectly aligned, and those can work pretty well. And so we have electronic monitoring is currently in place on about 10% of the fleet in New England, and there's a few boats out of Point Judith that are currently using it. And one of the fishermen here, pictured here, Chris Brown, he was using electronic monitoring, realized he had quite a large amount of data, of his own personal data. And one of the things that came about as part of this electronic monitoring was that the realization that electronic monitoring really turns fishermen's observations into hard numbers. You often go to a council meeting and someone from the stock assessment will say, oh, this is the numbers we get, da 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 da. And you know, the fishermen saying, oh, you know, we see thousands of fish, we see hundreds, we see fish here and there. And often it's tough to put those observations into numbers that can go into assessment or directly into management. But with electronic monitoring, we're getting hard numbers that are verified by people who are looking at the video, and we get that those numbers can go directly into research, stock assessments, management, that kind of thing. And so what we wanted to do here was see if we could actually use just a short time series of electronic monitoring data and actually see if we could use it to influence management. We go to the next slide. So this was a question about window pane flounder. Window pane flounder used to have a small fishery that they're relatively small, thin fish. So the fishery started to decline in the 1990s. And then in 2010, the stock was a little in trouble, so they banned fishery prohibition on the stock in general. And then the assessment is done through just looking at the index from the Federal Trawl Survey. And so the only real gauge they had was they would set a quota for how many fish could be caught and then discarded. And if that threshold was hit, I forget if it's 1 or 2 years in a row, then they would close particular areas where they thought there was a lot of window pane. And so these are called the accountability measured areas. So you can see in the top map here, just south of Rhode Island, right around Block Island, there's one of the accountability measure areas. And that is also a common place for many of the day boats out of Point Judith to fish. So it's a very important area. It's a good fishery. People spend a lot of time there. If we want to go to the next slide. So a question arose in, I think it was 2015, the threshold for window panes was hit. And so there was a concern that the accountability measure areas would kick in. There'd be a lot of restrictions in this area. As you can see on the right-hand plot with the orange box there is would be the area. And so there'd be additional restrictions put on fishing in that area. And like I said, it's an important fishing area. And so you can see here in the plot, these are the locations where a lot of fishing was taking place. And so some of the fishermen, especially those who had electronic monitoring on their boat, were wondering if this accountability measure area was actually in the right place. The way the system was working at the time was that they were trying to record regulatory discards. And so the catch would come up, they'd dump it into the checker, they'd sort all the target fish, and those would go into the hold and things. And then all the regulatory discards would actually go on the measuring board, as is done here. There's a camera above, and so each one went on individually, and then it would get thrown out of the chute. So the people on board had a very good idea of exactly the size and number of discards they were throwing over. Certainly, when you're shoveling fish overboard that don't make it, you have some idea, but when you're individually handling every fish, you have a really good idea. And so if you look at the plot on the right, you can see these are the number of discarded window panes, approximate. And you can see that the highest number of window panes are kind of right on the edge on the eastern side of that box and sometimes outside of it. And so Chris Brown in particular was talking to, I forget if it was Connor or Jason at DEM, and was curious if this electronic monitoring data could be used to see if that box, if that accountability measure area was in the right place, if this needed to be tweaked or how it could work. And there was also a little concern that this AM area had been put in place well after the fishery had been established. And so it was never really communicated very well from GARFO or from the council down to the fishing fleet about why it was there, what exactly it was, and what data had been built on. There are certainly concerns or thoughts were, if they wanted to protect window pane, they would have put this box where the fishery was. And the fishery back in the day in the 90s, in the 70s, 80s stuff, was actually much further east than where this box was. And so there was definitely, just general apprehension about why it was there and what it was doing, and then a concern that it could seriously impact the fishery if these accountability measures went in effect in like 2016, 2017, 2018, that kind of time period. And so that's what prompted this study, was whether we could look at the EM data as well as other data sets and see if we could just see what was going on, if we could make any heads or tails of whether this management area was in the right place or not. We want to go to the next slide. So we decided to try and look at all the data that was available in this area. So here, this is the southern New England down to mid-Atlantic. So this is the whole distribution of southern window pane flounder, at least as the stock is defined by the stock assessment. And so we took a number of fisheries independent trawl surveys that were built for identifying trends and distribution of window pane. But we also wanted to obviously include the EM data. We wanted to this really nice data set generated by the fishermen and use their data and integrate it into this study to make sure that we were directly including their observations in the study. So, and also they had the most amount of information in the study, the specific study site. So we combined the NEMAP study, you can see that in the pink on the left end, covering most of the coastal area, the federal trawl survey from the Hangman Bigelow in the purple, covering most of the shelf. And we have the DEM data in black, that's a little more focused. We have the Block Island wind farm data, and then we had the electronic monitoring data in green. So we were looking at specifically the years from 2010 to 2017, and maybe 2018 in the end. And so that was after the Henry Bigelow came online, so we didn't have to worry about conversions and things like that. And then we were looking at January through May with the idea that most of our the fishing fleet we were interacting with was really doing this in the spring, and then they would switch over to squid once the squid season started. So we were looking really at that January period. And so the way we combine these was using a spatial delta generalized linear mix model. And we use an R package called VAST. It's a very nice package. It really did a great job. And so if anyone wants me to go more into the methods there, I'm happy to talk that up quite a bit, but I'm not sure it's really needed. The important thing about it is that particular model, it allows for spatial autocorrelation, so you're able to account for those measures. And most importantly is that where the data are sampled in similar time and space, it allows you to understand the differences in the catch and attribute those both to potentially the environmental aspects and then also to the difference in the surveys. So you can actually get a catchability coefficient out of this particular model. So that was a really nice feature that we thought was very valuable, and I think it worked pretty well. So we were able to combine all these different surveys within that model and get good results. If you want to go to the next slide. So this is the output of that VAST model. So we were looking for the distribution of window pane flounder in the southern New England and mid-Atlantic Bight area. The model also produces index of abundance, and that was a little bit variable depending on what assumptions we put into the model, but the distribution was very consistent no matter what was very robust in the assumptions we made. And so on the left-hand side, you can see kind of the full picture of a good portion of the Northeast Shelf. You can see there's hot spots of where window pane are. The one thing is that to make all the data comparable is we only looked at window pane that were greater than 22 centimeters. That's what gets caught in the commercial nets. And so we eliminated all the juveniles. And so the picture would look different if we included the juvenile data. It would highlight a little bit more along the coast of New Jersey and Long Island. But the hotspots really are kind of off the coast of New Jersey, a little bit along Long Island, and then to the east of the accountability measure area, as you can see from the small plot on the right. And so what you can see is that there's window pane around for a bunch of areas, but the real hotspot is really to the east of that accountability measure area. And talking to a few of the fishermen, they had definitely kind of had the impression that there was that the window pane were generally to the east of the accountability measure area, and that depending on the season and conditions, sometimes they would expand a little bit west and get into their fishing grounds. Sometimes they would retract a little bit and they wouldn't catch many window pane. And so our distribution generally agrees with that idea. You can see that it really highlights that it is right to the east of the main fishing grounds where the accountability measure area is. But what we came up with the model was definitely a little bit deeper than a few of the fishermen said that they had traditionally caught window pane at. They seem to think they typically, when they would fish for them back in the fishery in the 80s and 90s, they would have been a little further north, a little closer to like Martha's Vineyard and Nantucket in shallower water. So they were a little surprised that we found this hot spot here in a little bit deeper water. So in general, kind of or similar to what the fishermen had anticipated about where the population was, but not exactly the same. But we did match up very well with the observed data, and we were able to match those pretty well. So what we found is, kind of in agreement with what the fishermen thought, that high abundance areas of window pane were not in the main accountability measure area. So if you want to go to the next slide. So we pulled in Ben from Garfo to take a look at the accountability measure area and in relation to why it was exactly where it was, and to look at discards of window pane. And so what we have is basically broken down by 10-minute squares across the whole southern New England and mid-Atlantic Bight area. We have, that's the plot on the right, a number, I think this is pounds or numbers, I'm not positive, of discards of southern window pane flounder. And what you can see in the red is that the majority, almost all the discards are right there in that accountability measure area. So what we found is that while the high concentration of window pane were not in the box, where the discards existed was in the box. And so we kind of showed that both the captains we were talking to were definitely correct about the abundance, but also that the management agency, was actually instituting things what they thought was in the right place. And the basic idea is that if you wanted to protect the highest abundance of window pane, you would have put the accountability measure area in a different place. But the actual fishing effort in that location is relatively low. And it might be, I'm not sure how big the screen is there, but if you look on the right-hand plot, you can see that basically everywhere it's white, there's no window pane discards. Where it's blue, it's relatively low. And there's not that much discards outside of those really large red blocks there. And those red blocks are right in the AM area. And so if you want to have an impact on reducing fishing mortality, you have to go where the fishing mortality is happening. And that's why the accountability measure area was put in place where it is. And so this information was never really conveyed to, I think, much of the fishing fleets. We talked to a few of the captains, and they were both, they were at least could understand why it was put in place there. So that made more sense to them. They weren't happy about it, but at least they understood why it was in place. And it also was a nice agreement that, they showed that their observations that they weren't crazy, that they, certainly what they were seeing on the water was that the main portion of window pane were to the east of this accountability measure area. And so we finished up this study. We were able to publish it this summer, which was pretty exciting in Fisheries Research. And we hope that while it didn't create any great headway, it didn't change management directly, it did show how certainly the fishing community and management might have very different perspectives of what's on the water and why things might happen. And so certainly communication between those two is very useful. And also combining data sets so we can create a data set that everyone trusts, that people, it's verified data that both scientists and fishermen can believe in and know that their information is in there and doing well. It's a good way to start, having that conversation to be able to move forward and have people, collaboratively working together as opposed to kind of being at odds with each other all the time. So with that, a quick conclusion slide here. So one thing we really showed is that electronic monitoring data, it was a really high quality data product. And so instead of just being a way to do compliance or something like that, it could actually be a data stream in and of itself. It's really a way to turn the fishing captain's observations into real hard numbers that can be directly input into quantitative analysis. And so we were pleased that this was, a fishery industry generated question leading to a research project. And we were able to answer their question, maybe not quite as timely as they wanted, or maybe not exactly how they were hoping. We didn't massively change it, though we could tweak the boundaries of it a little bit. But it was certainly, we were trying to get their question answered. And as I said, the density of the window pane main density was outside of the AM area, but the main discards were in that area, largely because that's where the highest effort is, and that's where the main fishing grounds are. And so if you want to reduce fishing mortality, if you want to protect the stock and have an impact in that way, then you need to reduce the discards. And so protecting where the high density areas, it wouldn't actually do that. And so with that, I'd like to thank a number of groups and people for being part of this. If you want to go to the next slide, and I'll take any questions. And if people ask questions, I'm going to ask that whoever's right next to the computer, if they repeat the question, because it's tough to hear it from in the room. Thanks.
Right,
Couple
Al?
of questions. Maybe I misunderstood what you were saying in the beginning of this speech there, but I thought he said they were only measuring 22-centimeter fish, the fish that the draggers were keeping, and the juveniles weren't being measured, they were being discarded. So my confusion is, majority of the discards would be in the area of the box where they close it off. So maybe you could explain what they're calling discards and how they assume that they were discards. I'm confused there.
Yeah, so hopefully I'll get this question right, but could you elaborate a little bit on how they were classifying or what is meant by a discard for this study? Or what?
All right, thanks. Yeah, sorry. Yeah, I was a little unclear about that. So window pane cannot be kept today. It's a prohibition on window pane. So any window pane that gets caught by the commercial fleet has to be thrown overboard. When we looked at the commercial data, they were only catching, because they were using 6-inch dragger, 6-inch mesh from the dragging gear, they were only catching fish that were 22 centimeters and larger. So if they caught smaller fish than that, those would also have to be thrown over. But they generally, I think there was one 8-centimeter fish, I think, in the entire pool of a couple thousand fish we measured. So that's why everything was considered, every window pane was considered to discard regardless of size. It just happened that the commercial fleet was only catching larger fish, but they used bigger mesh. And so we used, for all the data, we just used the larger 22-centimeter cutoff. When we looked at this, we did look at it using the juvenile data, and the pattern was relatively similar in that there was not a high concentration of window pane in that accountability measure area, but there were a few other places like along the southern coast of Long Island that did have a lot of smaller window pane.
Okay, thank you. I get it now. All officials consider discards. Yeah, okay, thank you very much.
Quick question. Thanks for, this is Connor. I had just two quick questions. The first is, I know that the program with the 3 captains from Rhode Island was funded, I believe, by TNC. Is that program continuing? Like, are we getting additional years of data? Can you speak to like how this analysis showed, like, help maybe continue the funding stream for that and the data's application?
So that is a great question, and I should know the details of that, but I don't know all the details. I believe that there was, I think it was a 3-year program, and then I think what happened was that the video cameras and stuff that were installed on the boats then became property of the captains of the boats themselves. I'd have to double check on that, but that's what I think happened. And so this was part of a larger project throughout New England to get video cameras on boats and to see how effective that would be. So what happened is, working with Garfo, anyone who had, once you had the EM system set up, then that would take the place of the observers. It wouldn't take the place of the NEFOP observers. It would take the place of the sector observers. And so the NEFOP would also be the ones that would collect biological samples. So they wanted to make sure that that was still there. But the regular observer coverage could then be done through the EM data. So they were submitting that with the regular way that observers would submit their discard data. A number of the captains elected to have the video cameras running 100% of the time. So they were basically 100% observer coverage. And so that gave everyone a lot of confidence that what we were seeing was really their active, real fishing activity and not, potentially biased because there was an observer on board or something like that. And so that is moving forward. It helped. This was part of the larger project and also part of Amendment 23 with part of the industry monitoring and observer aspect. And so, I mean, I don't know if we want to go into that forever, but this was part of that. And so certainly Garfo is moving forward with trying to increase observer coverage and including electronic monitoring as another alternative or option for moving that forward.
Thanks. And I guess my second question really quickly is, you talked about how the data, as you combine the surveys, both from the captains and traditional surveys together for a unified abundance for the area, you can also then produce annual abundance indices for the stock over the region. I guess I was curious if, I believe Southern Window Pane or Southern New England Window Pane Flounders assessment index based, and I guess I was curious if there's been discussions of how this work could be used to complement or in unison with the current index approach that the assessment scientists are using for the stock.
Excellent. Thanks, Connor. Currently this stock is assessed by simply using the index from the Federal Trawl Survey. So that doesn't cover the intro waters that would be from NEMAP. It wouldn't include any of the state level data like DEM or anything else. And so this method was specifically designed from on the West Coast to actually be able to take multiple vessels and combine it into a single index. That's where it gets its origin from. And so what we did here is we laid the groundwork for that. We were really focused on the spatial distribution, which produced a good result there, but it did also produce an index of abundance. And so I have Jason, I think, reached out maybe to the Groundfish Committee. I've a while ago, I did talk to the Groundfish Committee that we were doing this work. I haven't followed up recently. And I did send a note to Tony Schnuty, who's the lead assessment scientist for this, saying that, we put this model together, and so that if she was interested, we could certainly connect with her on that. As always, the assessment scientists are pretty overbooked, and so haven't gotten a lot of feedback from that as well. But the vast model that was used here is getting expanded use in the Science Center in general. So I know Kirsten's testing it out for, I can't remember which species, but so he tested out for a few species. There was some work done on it with fluke, and so it's getting more traction and potentially would be used. So I'm hoping that our base model could help out and inform the window pane in the future, and just the inclusion of more data in this type and accounting for all the spatial autocorrelation, getting all the state data integrated into the assessment in kind of a really nice framework. Thanks.
Thank you, Rich. Jeremy here. A quick question about the biological data. Are you able to get length frequency distribution from the observer data?
Were you able to hear that at all, Rich?
Not really.
So you're asking
Repeat
me to get length frequency data out of the observer data?
Sorry, can you repeat that one more time?
Yeah, were they able to get length frequency data through the observer data sets?
So for this, for our analysis, we did not have length frequency data from the observer data. So we had all the length frequency data just from the electronic monitoring data. So which would have been, some of that would have been what would have gone into the observer data set anyways, but we weren't able to make direct comparisons there. So what we were thinking about, however, for fluke, they've been recording these length frequency data sets for all the regulatory discards. That's just part of the program. And so they've been collecting a lot of fluke data, including a lot of juvenile fluke data. And so we had discussions initially, we haven't had time to follow it up, but then like talking to Mark Cicero and seeing if we could, have this small subset of data that just around waters potentially compare the length frequency data of these discard data, especially for people who are recording 100% of discards, seeing how that matched up with the discard data he's getting from the larger observer program, see if they were similar or different, and potentially exploring if there were differences, why that might be, and if we could see more patterns or more detail from these EM data compared to what they're getting from the more general observer data. But that's another idea and study yet to be done, but it's certainly in the back of our heads to try and get that done.
Not picking up myself.
Okay. Rich, can you hear me now?
Yeah, I can hear you better, yes.
Julia, turn the microphone on. So your results imply that the fishermen who are fishing in the accountability area are targeting something else. What are they targeting?
Yes, that's a very important part of this whole thing. So most of them were just ground fishing at the time. So general groundfish and also including fluke in there. And they were not targeting window pane. That's very important for some of the assumptions within the model. So definitely plenty of flounder species in there. And I doubt they're targeting any cod or haddock down there, but I think flatfish and a few other groundfish species were the main target. And so one of the important assumptions as part of this was that the window pane were not being targeted so that we could consider them a random sample, and also that they were not being avoided. And talking to the fishermen, they said that they would just fish normally, and then only when they get really close to that threshold do they even think about window pane, and potentially trying to avoid it, or maybe fish with the net a little bit lighter, a little less on the bottom, so that because they have the impression that window pane are the most sticky to the bottom. So if they fish a little bit lighter, they can go over them a little bit and catch some fewer. So that's what. But so that was an important consideration that we made sure we checked with all of them, and they said that they don't target them or avoid them in general, and they just go fishing and then kind of hope that they stay under the threshold.
Any other questions? I'm not seeing anyone online.
Great. I guess, so with that, kind of wrapping up the talks then for today, I'm first just gonna say thanks everybody for attending online and in person. I want to remind folks too that in a normal year, we typically try and have 2 research meetings, one in the fall and one in the spring. So keep an eye out for the spring. We talked also about perhaps a theme meeting for the spring that focuses on Narragansett Bay and the ecology of various aspects as of interest by various stakeholders, as well as some of the emerging research that's come out over the last few years to try and capture that a bit better. So if you have ideas or interest in future theme sessions or talks, please let us know. I also want to say that a lot, as Rich mentioned here as an example, a lot of the research that we do comes out in part based on the discussions we have between the agency, the universities, and various stakeholders in the commercial and recreational industry. So for those, I often look at RIMFI as a think tank for trying to capture these ideas for work that we can pursue in the future that's endemic to around fisheries. So for those of you that have ideas on certain research directions for the Institute, things you'd like us to see and pursue, please feel free to reach out to Jeremy or I, and we can continue to keep those on our docket for ideas. So when opportunities come up for funding, we're able to respond swiftly. So, Jeremy, do you have any other comments you'd like to share?
I would actually, I think you got some very good feedback this afternoon. So it wasn't that, and we'll be following up on that. And also, we do welcome new ideas for new permanent projects. Thanks for attention.
Jeremy, I got an idea right off the top of my head for a future project. An in-depth study on shell disease. It was done some years ago. Kathy did it.
Yes.
But shell disease is getting more rampant now than it's ever been. It's worse, getting worse each year. I can see all the research I'm doing for the foundation. My dental scratch studies. And I just think that something really needs to be done, investigated into this. I can approach it, but it's getting worse, and it's taking a detrimental hit on the fishery, especially with the Jaegers. It used to be with the Jaegers, we get severe shell disease by springtime, just prior to releasing their eggs. Now, I'm noticing there's severe shell disease even in the late fall and early winter. And those Jaegers aren't even going to live to hatch their eggs. So it's a real serious situation. I think it's something, a project that if it could be looked into and something studied to be done, would be very helpful to the industry.
Do you feel that the incident is related to water temperature?
I have no idea what it's related to. I don't believe so, because it's progressively getting worse. Water temperature offshore is actually warmer than in the bay, and offshore you can see very small incidents of shell disease. So basically focused in the bay as you move offshore, even though in the area too, there isn't the incident. So it's a little concentrated there against the bay. So I think it's something that really needs to be looked at into.
Good point, and point taken. I mean, I think the previous studies were a little bit frustrating in that they weren't able to come to a definitive conclusions. But that's not to say that new approaches couldn't be used.
Thank you very much. That'll be my recommendation. I am the recommendation from a fisherman's perspective.
Now, not to put you on the spot, but some of the things we also hear about are like increasing or emerging fisheries in the bay, and then species that we may not have a lot of life history information on, or understanding how they interact, or those species interact with the bay's environment. So I think about conch, I think about mantis shrimp, think about these species that we have very little biological information on. I didn't know if you care to comment on maybe ways that the Institute could improve our understanding of those resources for an effective understanding of how the stocks are doing.
Yeah, the only approach I think you can take, the only people who have any data on all is the fishermen themselves, especially in regards to the mantis shrimp. I mean, that's a pretty big fishery these days. There's no regulations implemented on them, so it's a question how long how long biomass is gonna last, so if you really want to get a handle on what's going on, interviews like Sarah's done with fishermen, and ask them, what's been going on with fishing, what they see.
Great, thank you. Appreciate that.
Yeah.
Obviously, it's a group that's interested in fishermen's observations and Narragansett Bay. So I have a treat in store for you guys. Next Thursday, a week from today, it's December 9th at 4 p.m. Narragansett Bay Estuary Programs hosting a webinar about a project that I did where I interviewed 17 commercial fishermen in depth about all of the changes that they're observing in the bay from top to bottom of the ecosystem. I put that together into sort of a pilot project framework that we'll hopefully be able to build on all together to really give those observations and the fishermen who contributed them a role in ecosystem level management of Narragansett Bay. So if you, it should be on the website of the Narragansett Bay Estuary Program on their events or their calendar page if you haven't received an invitation, or if you know how to get in touch with me, Sarah Schumann, please reach out and I'll make sure you're on the list. Thanks.
Question, sir. Any possibility that they could do an in-person meeting? It's all gonna be done.
I can do a small watch party, if only with fishermen if there's a need. So let's talk after.
Thank you.
Any other comments there you see?
No, I'm just trying to relay. I don't think people can hear super well, so I'm just trying to relay what both of you were saying in the chat.
All right, I guess with that then, thanks everybody for attending and we look forward to seeing you in the spring, if not sooner. Thanks.
Thank you very much.
Yeah. Yeah, that's kind of managerial, but I mean, it's